I think we can go ahead and get started. But thank you all so much for joining us today for our Knowledge Portal Network webinar series. Oops, I forgot to update the date on that. But we just want to kick off with some upcoming events that will be going on in 2025 as we're nearing the end of the year. We just want to have some dates that are held on your calendar for 2025. And so the first one is going to be our Common Fund Data Ecosystem virtual workshop. And essentially, this was something that sparked from ASHG this past year. We had such a successful session in our ancillary session that we wanted to bring this to a greater audience, and we'll be expanding on that session that we already had. So this Common Fund Data Ecosystem virtual workshop will be geared towards human disease researchers and will focus on empowering discovery through open access data and resources. So this event is going to be January 31st from 10 a.m. to 2 p.m. Eastern. We have the registration link available. This is going to be through a webinar. And if you have any questions, please reach out to us at help@cfdeknowledge.org. And so to give you a little preview of some of the items that we will be discussing during this talk will be a featured presentation from the NIH with their perspective of the Common Fund Data Ecosystem as a whole. We'll have the Knowledge Center and Data Resource Center overview. We'll have a scientific presentation geared towards gene set analysis, and we'll have a few introductions to the new centers that have been joining us as part of the CFDE as a whole. We're going to also go over some funding opportunities. And as of this morning, it is confirmed we'll be doing a Terra workshop discussion, and this will be kind of the piece at the end that it'll be a little bit interactive if you're interested. So this is something that we're excited about, and we hope to see you all there. Again, if you have any questions, please reach out. We'll be publicizing this a little bit more with the registration link, but I will send that to everyone as well. So this is just a date to hold as it is upcoming. And as I said, this is our last KPN meeting for 2024, but don't worry, we're going to have lots more science coming, especially in 2025. So a few early presentations that we have that are definitely 100% on the docket are going to be the Mammalian Adipose Tissue Knowledge Portal, or MATKP. We'll be introducing this to our team here. And then we'll also have a presentation on the Nephrotic Syndrome Knowledge Portal, which is another dev portal that we've been working on for quite some time that we are very excited to share with everyone. So as a reminder, we have all of these booked up throughout the year, so please keep an eye out for them. And just a programming note, you might see some information from me coming out with our lovely colleague Maria, who is retiring at the end of this year. We actually will be switching over to a different webinar link. So if you see any updates, it's because of that. And we just want to thank Maria for all of her hard work and help with all of these projects. So thank you, Maria.
Thank you, Mackenzie.
Yeah. But with that, that's all the programming notes that I had, and I will turn this over to my colleague, Julie Juergens.
I think I'm going to take a quick,
Never mind.
jump in. That's okay. I'm going to keep it really short so that Julie and Jason can have the full time today.
Great.
Yeah. So when you said that's funny listening, not giving the intro, Mackenzie, I realized that this is Maria's last webinar. I'm not going to get emotional, I promise. I'm going to save that for other times. But so much of what we have done here as a community is because of you, Maria, and your kind spirit and your knowledge and your overall presence as a human being has, again, been the voice, but also the part of the portal that would not be the portal without you. And I mean that. And I can say that because I've seen it all and I've watched you help us grow. And I'm really grateful for you.
Thank you so much. I'm grateful for all of you, and it's been wonderful.
So I did get emotional, darn it. Anyways, so many of you, like I see some of your faces who have been in webinars, so you guys know this has been, Maria's been around for close to 9 years. I hope she make a decade, but I couldn't convince her to stay longer, but that's fair. So with that, let me transition to our presentation. So I'm going to give a quick intro because I think, looking at Mackenzie talking about 2025, The types of diabetes knowledge portal will turn 10 in 2025, which is exciting, officially 10 years old. And that's kind of neat. And we started as a shop building only one little tiny portal that had 9 data sets across 25 traits. And now we're talking to you about a brand new initiative that we have taken on actually simply because of the type of work we've done with a lot of you who are listeners who I see today, who have contributed data to our early knowledge portals, who literally helped teach us how we think about building resources for others to use. What I mean by that is we think about these things in ways that we really try to take our time with you all to listen to what's useful and make a resource that is accessible to that particular need. And I mean that because we may not be able to do everything that we should be doing all the time, but we're trying to be very careful and think about, what does a human geneticist need when they are trying to study their favorite gene? So that's really where our knowledge portals started from. And what's really cool about this new initiative is we're actually able to take some of the best of what we do in human genetics and marry that to an entirely new ecosystem that is way beyond human geneticists' reach. And so I'll share my slide real quick. There's a slide that kind of makes me think about what we're trying to do today and beyond. Where is it? It is, here we go. Yeah. So today we want to guide you. The Common Fund data ecosystem is something you may have heard buzzed around, particularly because you may have heard of the Common Fund, but we want to make it more accessible to a group of scientists who may be studying a human disease or a biological process. I'm sure you've probably heard of the Common Fund. Julie's going to talk about that a little more. You probably know giant initiatives like All of Us, that's funded by the Common Fund. You may not know about the fact that GTEx is a Common Fund resource, right? But you probably use that every day in your life. So it's actually a pretty complicated and large ecosystem. And the ecosystem is a great word for it. If you're a biologist by background, know the ecosystem has species and components, and they all do their own part. They have their own function, but they leverage each other, and they are changed and modified and live and breathe based on the interactions with others. So I love that word. I think that word is part of how we think about the ecosystem, because we think we have a unique role in it, but we are changed and shaped and fashioned by all of these resources. So there are 18 data coordinating centers. Julie's going to cover those a little bit. And then 5 centers now funded to make these data, knowledge, results, missions of all of these resources more accessible to you, a human geneticist, or a biologist, or a computational scientist, or even a clinician, to make them more useful and allow you to understand, well, wow, I'd like to dive deeper into Senet, for example, because I didn't even know what it did. So GTEx, you may not have heard of, you probably used it many times in your life. So that's a classic Common Fund data ecosystem resource. You may not know Spark. I didn't know Spark when I first joined this. This is stimulating peripheral activity to relieve condition. This is an electrophysiology-based study to understand how are the signaling that happens in a different event in the body, and it creates anatomical maps of that. So like these reference maps of signaling for electrical stimulation in the body. If you're a biochemist, you know this one, right? You know about glycobiology. This is a resource that's been around for close to 10 years now that generates all these results for glycoscience. It's a huge data repository. It has a large consortium around it, but how is it useful to you, the average person who might be studying a disease or a trait? Well, our job within the Knowledge Center is to help figure that out. And so with that, that's really what you're going to hear about today. You're going to hear about how we connect those things, because our approach is really about presenting results from these Common Fund resources to those who study a human trait or a biological process at large. We aim to support discovery. We're not going to necessarily discover things for you. That's your job, but we're going to give you a vessel to support that discovery, to generate hypotheses, to think about it. And we do this by integrating across biological resources. And what's great here is you're going to hear about the complement of all the work we've done, and I shared this with you over many years, in the complex trait space to build the first diabetes portal, and now expanding to the common metabolic disease knowledge portal, and then to the association to function portal, where we have 1,200 traits across all different disease areas, mapping that to what's in the Common Fund data ecosystem for commodities for resources. And I'm going to stop there because I think Julie and Jason are going to talk very specifically about that commodity, things that are produced by all of these resources in common. That's something that's very tangible and useful and and easy to connect to human biology and human traits. So with that, I'm going to pause and I'm going to hand it over to Julie.
All right, thank you, Noelle.
Just one thing before we get started, just a reminder that you can please use the chat or the Q&A if you have any questions or comments. We will be monitoring these throughout, and please feel free to do that. So thank you.
Yes. So as Mackenzie said, we want this to be interactive and useful to all of you, but our goal today is to really introduce the Common Fund Data Ecosystem, or CFDE, and describe how you can use it to get open access knowledge of these programs, analysis tools, workflows, and various resources that can help accelerate your research. So we're going to start by talking a little bit about what the CFDE is, how it's funded, the programs within it, and then how you can navigate your way around. To start with, within that scope, we're going to talk about the NIH Common Fund. So as the name suggests, this is a funding mechanism that supports collaborative initiatives that are not just within a single NIH research institute or center, but span multiple institutes and centers. And the benefit here is that these can really catalyze transformative discovery and address big picture challenges that face multiple branches of the NIH and really accelerate research. So these programs that are funded by the Common Fund are designed to be sort of time limited in their Common Fund support. So this gives them a kickstart, and then they can go on to sustain themselves through other funding mechanisms in order to keep going the resources that they built. So the NIH Common Fund's major goals are to accelerate emerging science, remove the big roadblocks that exist for research, build up the research workforce, and to support sort of high-risk, high-reward science. If you look up what the NIH Common Fund programs are, as Noelle mentioned, they're quite diverse. You can see there are programs that are currently funded as well as previously funded archived initiatives shown here. And a subset of these are integrated into what we call the Common Fund Data Ecosystem. So the Common Fund Data Ecosystem, or CFDE, promotes the accessibility, use, reuse, and integration of multiple data and resource initiatives from the NIH Common Fund programs. And it not only brings them in individually, but brings them into a cohesive ecosystem so that you can connect these resources built by multiple programs. CFDE also aims to do training, outreach, to promote awareness and to teach people to use these resources. That's a part of what we're doing here today. And also aims to build up the workforce. CFDE is supported internally by 5 major centers that have major deliverables, I guess you could say, in terms of what we're aiming to bring the community and how we're aiming to help users to use CFDE data. So we are representatives of the Knowledge Center. As our name suggests, we want to transmit knowledge about how CFDE works and develop analytic tools so that users can obtain new knowledge. We have a sister resource, the Data Resource Center, where users can go to get data from CFDE programs. New initiatives, the Cloud Workspace Implementation Center, which aims to help users to access data in the cloud, and the Training Center, which is developing training resources for users who wish to interface with CFDE. These efforts are all brought together internally by an Integration and Coordination Center. So I just talked about a lot of different things with somewhat similar names and somewhat a bit of overlap. So I wanted to sort of summarize what those are and how they all fit together. So the NIH Common Fund funds Common Fund programs. You might see those called as Data Coordinating Centers, or DCCs. A subset of these programs are integrated into the Common Fund Data Ecosystem, or CFDE, both as individual programs and as networks of interconnected resources. CFDE is supported by these 5 CFDE centers that I just talked about. So as one of those centers, we, the CFDE Knowledge Center, aim to improve understanding and visibility of CFDE resources. We tried to generate knowledge by analyzing CFDE data and creating a user-friendly centralized entry point for people to come in and interact with the ecosystem. It's designed to be integrative and, importantly, disease-focused to accelerate discovery. So what can you do through the CFDE Knowledge Center? Two major things. First, you can learn about CFDE, and secondly, you can analyze CFDE data. So to learn about CFDE, we have a number of programs, one of which is our research webinars like the one today. We also have workshops like the one in January that Mackenzie just mentioned, and events at meetings such as the American Society of Human Genetics, where we talk to people and put on ancillary events to help them learn about CFDE. We also have program information represented on our new website, cfdeknowledge.org. So you can go to this website to learn a little bit more about each specific program. Let's say you want to learn about this program MotorPack. You can start by hovering over it here within this schematic, or by clicking through the menu here at the top. And that'll take you to a page that looks something like this, giving you an overall sense of what the program is, a link to it so you can explore ideas of how to get to the data and the types of data that are represented there, an overview video, and then capabilities and knowledge generated by that program. Noelle gave you a flavor of what some of these programs are before, but I just wanted to give you a broader sense of the programs that we currently have represented on the CFDE Knowledge Center. As you can see, these span a wide range of biological processes. And because of that, you can really ask innovative questions in terms of tying together very distinct forms of biological research. To give you some ideas, we have down here on the bottom Glygen, which studies protein glycosylation, or the attachment of carbohydrates onto proteins to regulate them. xRNA, which studies extracellular RNA and its role in basic biology, but also as a potential biomarker for various diseases. Metabolomics Workbench, looking at metabolic signatures within various basic conditions and also disease. The 4D Nucleome Program, which looks at chromatin structure and epigenomics. Two initiatives to look at cellular maps of the human body. Those are HubMap, creating the first, sort of body-wide cellular map of the human body, and also Senate, which is doing that for a subset of cells called senescent cells. The GTEx initiative, looking at gene expression and regulation, which Noelle just mentioned. The LINKS program, which studies cellular and expression signatures in response to various perturbagens. So these can be knockouts, these can be drugs, these can be various microenvironments, and how those affect various readouts. This is a newer program, A2CPS, acute to chronic pain signatures. So a subset of people who develop acute pain go on to develop chronic pain. And this program is looking at what the signatures are that identify that transition. The SPARK program is looking at the nervous system with a focus on the autonomic nervous system, as well as bits of the peripheral nervous system in general, and brain-body connections. And the ultimate goal here is to actually do neuromodulation. So to think about how we can tap into the circuits identified through this resource for future therapeutic intervention. IDG is illuminating the druggable genome. This program focuses on drug development for specific classes of sort of understudied dark proteins. These include kinases, GPCRs, and ion channel proteins, which have historically been quite druggable and successful in therapeutic targeting, but some of which are understudied. Finally, we have MotorPack, which is looking at the molecular effects of exercise. Gabriella Miller-Kidd's first, which is looking at rare pediatric disorder genetics for both structural birth defects and pediatric tumors. And Bridge to AI, which is another newer program looking at artificial intelligence and how we can integrate it into biomedicine, not just through creating programs and tools, but also how we can create new workforce standards, ethical guidelines, and ethically sourced data sets to do that in a responsible way. So what are the resources that have been generated by these programs and how do you access them? The first is multiomics data. So you can find out what types of multiomic data exist by looking at our homepage. You can hover over a specific type of data, such as transcriptomics, to see all of the programs that have that represented. And you can also go to each individual page for these programs to see additional data types. So Spark is an example of one that has a few types of omic data, but their specialty really hinges on other types of data like microscopy imaging, connectivity mapping, and development of code and computational tools and maps. So the second major thing you can do through the Knowledge Center website is analyze CFTE data. We have a couple of ways that you can do that currently, and I'm going to go through each of those. So you can start by searching your favorite gene. That'll come up with a dropdown that looks something like this. You can click on this to get to the gene page, and that will give you examples of the types of analyses that you can do within existing programs for which we've integrated data. We have a few programs currently represented and are actively bringing in more as we go along. So for example, here you can see whether your gene is expressed and or has tissue specificity in the GTEx project. You can do similar sorts of analyses for phenotypes. So these are examples of some of the phenotypes data that we currently have represented. You can also look at differential gene expression analyses. So there are two ways to get to this analysis from our homepage. You can go to this icon if you scroll down, or this sort of drop-down menu up at the top. And differential gene expression analysis basically allows you to identify putative biomarker genes whose expression differs among tissues or phenotypes. So you can search by a gene, a tissue, or a phenotype and explore within that. We currently have transcriptomic data represented from the GTEx project and from Kids First, and we're working on bringing more. So this is an example of the types of results that you get here, visualized as a volcano plot with a table of results down below showing differential expression. The second type of analysis you can do is analyzing for gene sets. So I'm not going to go into this too much just yet because that's the focus of the next part of the talk. But you can get to that similarly through our homepage. And this analysis allows you to perform cross-program analyses of gene sets and how they relate to additional genes, phenotypes, and mechanisms. So coming soon, these are some new initiatives that we have. We're working on developing a CFDE Terra workspace. We're starting by bringing in the differential expression and gene set analysis results that we just talked about and are actively building this out. So we're going to be talking about this more during our workshop in January, and we're excited to tell you all about it. We're also working on developing a single cell browser where you can explore single cell RNA sequencing data and look at expression by UMAPs, by lin plots, cell counts, marker genes, and other features. We're bringing in data from Gabriella Miller Kids First, which allows you to look at counts of variants and their consequences for various phenotypes, as well as transcriptomics for genes that are up or down regulated in response to exercise in rats from MotorPack, and glycosylation data from Glygen. So this allows you to search for a gene and get a table of its glycosylation sites of its encoded protein and whether it encodes a glycohydrolase or glycosyl transferase. And finally, we're actively developing all these resources. So in the future, we'd like to bring in additional transcriptomic data sets, additional gene sets, and eventually also external data. And finally, long-term, we want users to be able to bring their own data. We're not quite there yet, but we're excited to bring this to you in the future. So I'd like to acknowledge everyone on our team who has contributed to this. This has been a multi-person undertaking through individuals here at the Broad, as well as our colleagues in Kyle Galton's lab at UCSD. So I'm really grateful to all of them for their hard work on this. So with that, I'm going to pass it off to my colleague, Jason Flanick, who's going to talk more about one of those analysis types that I just mentioned to look at gene sets. Jason?
Yeah, hi. And does anybody have, or do questions in between, or should I just go right into it and do questions at the end?
Do we want to do questions at the end?
So anyway, while I'm setting up, if anybody does anybody have any questions for Julie before, quick ones before I start?
I haven't seen any in the chat and Q&A, but you are more than welcome to ask while we go through if you're okay with that, Jason. But
Sure, great. All right, so hi everybody. So we're going to now take a deeper dive into the gene set analysis workflow that Julie alluded to. And that's because we think it's a particularly useful thing for people studying complex traits and hopefully in the new year, rare diseases as well. And I'll kind of motivate that and then kind of go through a deep little dive as to how you might use this. And we are actively looking for people to help us browse these data. I do think there's a lot of interesting stuff here. So I'm hoping we can at least spark one of you to maybe reach out to us after for some more details. That's kind of the goal. So the main challenge we faced and still face kind of coming into the common fund data ecosystem as human geneticists is how to wrestle with data that's kind of a completely different type than we typically work with in human genetics. And Julie covered a lot of that, so I won't go into that. Only to say that it's, layering on these kind of, in some cases, exotic and different and very heterogeneous data sets on top of this very, I don't say rigid, but very specific model we have of how genetic variants impact regulatory elements and genes and ultimately biological processes is kind of a challenge because not all of, many of these common fund programs don't really interrogate genetics or diseases or variants. So the way we have thought about this is that if you kind of just sketch out like what types of information do each of these resources have regarding aspects of this kind of variant to function workflow, don't take this as gospel, but a lot of them have information about genes and biological processes. That seems to be where most of the information that these data sources have. And so I think the question is, if we're looking at how these might be useful for human genetics, is can we kind of extract the information that's buried inside each of these resources and use it to give us information about genes and functions, in other words, mechanisms that might explain some of the findings that we're getting from human genetics. And since all of our portals are based on human genetics, we think that Common Fund can be a really valuable addition to that quest of trying to find processes underlying the disease state. So if you think about how you might do this, right? So obviously you can look up in the publications from the Common Fund whether they published a gene that showed up in your GWAS. But obviously that's like only a fraction of the available data, and you're missing out on all of the much larger set of information underneath that. Another approach that a lot of people take, and this is something that's being done in the CFDE as part of the data distillery, is to build these knowledge graphs where you represent genes as these nodes, and then you link out these nodes to other nodes that are developed from different Common Fund data sets with these assertions. And if you construct this knowledge graph like this, you can do this type of like machine learning or reasoning over it, and you can do queries like, here's a gene for my disease, what might it be involved in based on Common Fund? And that is something that we are doing, and we'd be happy to talk to you about this. This is another group, actually, Dan Taylor and Jonathan Silverstein, who are doing this within CFDE. And that's not what we're going to talk about today, but like I said, it is underway. Instead, we've been thinking, and this is part because, Avi Mayam's group who run, and Shankar's who do the Data Resource Center think in these terms, is a different way of connecting these type of data to genetics. I'm thinking of this as a different type of universal language that's more lightweight, which is gene sets. So all of you are probably familiar with gene sets, simply a list of genes that you generate from some type of project. And I'm showing here an example of one from MotorPack. which is a set of genes that were upregulated after 8 weeks of exercise in the blood of rats. And the nice thing about these is because it's just a set of genes, you can connect it to any other type of evidence source that also produces a set of genes, like human genetic studies. And the good thing is that there is tons of these that have already been generated by CFDE. There's about 37,000 gene sets that have been generated via a variety of methods from these Common Fund programs. A lot of them are based on differential expression analyses, like the one I showed for MotorPack, but there are others like from links that are genes that change regulation in response to, a chemical treatment or a gene knockout. And then you can also do things like intersect these genes to find gene sets, to find pairs that kind of reflect both of the processes being interrogated. So you can think of this as like a ton of information that kind of tags different biological processes being interrogated by these Common Fund programs. Now, the issue there is because they're so easy to generate, you have so many of them, and it's not obvious just by looking at the gene set what it's interrogating. The name tells you something, but only part of it. And moreover, it's not at all obvious which ones are relevant to a trait that you might be interested in, right? The vast majority are irrelevant. And so the idea is that we need to apply some type of method that can filter through all of these different gene sets and cross-reference them with a human trait or all traits in our portals of interest, and then try to find only those that are relevant to traits of interest. So that's basically the idea behind this gene set analysis workflow, which is to essentially combine these gene sets generated by the Common Fund with the human genetic data we have in the knowledge portals to try to link processes tagged by the CFDE to human traits. And if you have been following our work here, you may have heard of Pigeon. You won't be surprised that this is the method that we're going to use. We presented this on a webinar, I think in June. That's available on the homepage of our portals, and you can go to there if you want to get full details on the method. I'll give a brief review here and how it's relevant to this problem. And you also, there's a web portal that you can browse if you want to try to see it applied to the traits. And please reach out to us if you're interested in learning more about this outside of the context of CFDE. So the main reason, the main use case of Pigeon for CFDE is kind of to produce more interpretable coefficients than we get for Magma. So if you're familiar with Magma is like kind of the common way that most people link gene sets to a human trait. Kind of convert all your GWAS to a list of gene scores, and then you do some enrichment analysis. But the coefficients are very hard to interpret, and overlapping gene sets are all very highly associated. And you get these biases such that larger gene sets are more enriched. So the main benefits of Pigeon, that we're using in this specific setting is that it produces very interpretable and sparse coefficients. And I'll show an example of exactly what that means momentarily. Just a recap of how this method works. It's a joint probabilistic model of gene probabilities for relevance to a trait and gene set associations relevance to a trait. And they have all this, these complicated probability distributions that we put on the values, which I'm not going to go into today. I'm just going to give the intuition as to how it fits the model and then kind of what it looks like and why it's a useful method for what we want to do with CFDE. You can think of what Pigeon does as it kind of guesses the gene scores. Like here, each kind of box is representing a gene. The red are more associated with your trait. And let's say you guess those from Magma or the huge scores that we have in our portal. And then genes are grouped into these gene sets that can overlap in the circles. And based on your initial guesses of the gene associations, you identify the gene sets that are enriched. Those re-update your gene scores, and then you identify additional gene sets that are enriched, kind of go back and forth until you get these joint estimates of both the gene and the gene set associations. So basically, the most important thing to understand is what this produces is for any given trait, you get the probability that each gene is relevant to the trait, and also the effect that each gene set has on that probability. Okay, and so like it's easiest to see with an example. If you run this for type 2 diabetes with gene sets from like text mining and mouse phenotypes, the output for genes, you get 3 values. You get a value of the overall probability it's associated with that trait. That's this combined column. And that's decomposed into effects from the genetic associations directly for the gene, that's this direct column, and the indirect, which is coming from the gene sets. So these are typically of most interest if you're trying to dissect a trait, you want to know the gene scores. But for CFDE, we also care equally about the gene set scores. So for each gene set, you get an estimate of the effect that that gene set has on the probability. That's this joint effect. And that's in the joint model of accounting for every gene set at once. And then you also have this marginal effect. And that's actually that's the effect size of the gene set in isolation and is more typical to like what you get for an enrichment analysis and is most relevant in the context of where you're trying to evaluate whether a gene set is relevant to a trait. And so for CFDE, that's what we're most interested in. We're trying to take all of these on the order of 140,000 gene sets that we've generated, and for each one determine which of the 1,200 traits in the Association of Function Knowledge portal it's linked to. Okay, and I think this these slides kind of illustrate why Pigeon is a really useful model in this context. If you look at like the distribution of gene set effect sizes from POPs, which is a method that uses magma for a similar end, you get these like highly like, you know, tons and tons of gene sets that have some effect size, and they're positive and negative. By contrast, Pigeon has on the order of like 100 times fewer non-zero parameters, and all of those that are non-zero are positive. So it's much easier to interpret what's going on here. And you can kind of see this like the top associations for type 2 diabetes for Pigeon versus POPs. For type 2 diabetes, all of these are positive, and they're you could convert these to probabilities. Whereas for POPs, what you get are these effect sizes that can flip in sign, whether you consider the gene set on its own or together. So it's much, much harder to interpret like what like the gene set values related to a trait. Whereas because Pigeon is this model that allows you to be very sparse and probabilistic, they're just much more interpretable. And that's very critical when we are trying to, for each trait, try to find CFDE gene sets that are linked to it. Okay, and that'll become clear kind of when I kind of show you on this portal. And Julie, I think, will give a deeper demo, but I'm just going to dive in, trying to go deep on a couple examples so you can kind of get a sense for the type of information that this method's producing. So on the portal, if you have a trait of interest, the easiest way to access this is you type it into this single search bar. And then if you click on Open Gene Set Analysis Browser, I'm showing here the results for T2D. So this is going to correspond to that table I just showed, but that table I just showed is not using the CFDE gene sets. This one is. So by default, what this table is doing is ranking all of the gene sets across all CFDE projects that are associated with type 2 diabetes. Okay, and to go through one, actually, I think I'll show this second one, the top one here on this page. So in terms of information of what these values mean, you can hover over the table headers and they'll give you information about what's significant and what's not and what all of these columns mean. And so this is an example, this relationship of a gene set, C60 adrenal male 1 week up, GTEx adipose tissue. Okay, so this one is a gene set that is upregulated genes after 1 week of exercise in the adrenal gland of motor pack that are also downregulated in GTEx adipose tissue in elderly people. And the details of that are, available on the DRC and also in, we'll show in a minute on this hypothesis tab. And then what this is telling us is that this has an effect size of 0.07 with type 2 diabetes. And if you hover over this, you'll see that 0.01 is significant. 1 is strongly significant. So it's a relatively weak effect, but still significant. There's 23 genes in this gene set. This is the source of it. So it's telling you it's from the crossing of motor pack with aging. And then if you expand here, you'll get a list of the genes in the gene set. And these genes are ranked by the association with type 2 diabetes. So in addition to showing you that this gene set's associated with type 2 diabetes, it's also telling you which genes drove that association. So SEBPA, AdipoQ, all of these genes are the top ones. If you go over to like this third tab, you'll see there are also some genes in the gene set like FFAR4 and NAT8L that are not associated with that trait. That's what this combined genetic support is. So that's the genes in the gene set. You can click on these, and I think Julie might show more to go to these pages that will give you more information about the genes association across many phenotypes. But we're kind of focusing here still on the gene set here. Okay, you can click on the gene set and you can get a similar PheWAS plot that's going to show you other traits that it's associated with. So this gene set, in addition to be associated with type 2 diabetes, associated with adiponectin, random glucose, and HDL cholesterol. Okay, so in terms of what this is telling you, okay, essentially you can think of this as hypothesizing some link between these genes, okay, in this gene set and the phenotype type 2 diabetes, mediated by some process that's tagged by this adrenal 1 week up, whatever. Okay, and so you may not know what this is, but if you type this into an LMM, it actually can do a pretty good job of guessing like what this is measuring, because it knows what GTEx is actually in adipose tissue, and it knows these are ages and down, it learns it's down-regulated. So you can ask it a query like, how might these genes be involved in type 2 diabetes as mediated by this mechanism? And this hypothesis button, which is broken at the moment because we had a security breach yesterday, but will be up probably in the next couple of days, will actually pre-run that query for you. What it kind of looks like when it's working, it'll show you here on the left the query that it executed, which is basically some context surrounding how the gene sets were generated for MotorPack and GTEx. And you can see down here, this is the query. In one paragraph, how might these 10 genes relate to type 2 diabetes in the context of that gene set? And it will give you kind of its guess at a hypothesis. And I've actually found this to be a very useful first cut to kind of screen whether these results are interesting and give you any insight into your disease that you're studying. So in this case, what it says is that these genes are involved in various metabolic processes, dysregulate type 2 diabetes, down-regulating adipose. So it gives you some type of insight about, it's linking it to insulin sensitivity and lipid metabolism and insulin resistance. So that has been like a pretty good starting point to then kind of dig a little deeper into what mechanism this might be revealing underlying T2D. And so just to remind you, and I kind of have, I think, a couple slides on some examples of how this looks in practice, but just to remind you, so MotorPack they're exercising rats and seeing genes that are upregulated. So these genes are going to be somehow involved in exercise in the adrenal gland. And then this is adipose tissue down expression in elderly people. So you're having adipose, you're having exercise, you're having adrenal gland, you're having type 2 diabetes. Obviously, you can probably guess how there's going to be a link there. And others might know better than me, but some basic research based on that ChatGPT hypothesis. So most of you are probably familiar that PPR gamma, SEBPA, these genes are involved in adipogenesis and lipid storage pathway, and that's how we typically think of them. But the CFDE gene set on MotorPack and the adrenal gland actually kind of pulled up this role in cortisol response. So adrenal gland is responsible for cortisol release, and leptin apparently inhibits that, and leptin's in this gene set as well. And so it's kind of by having access to this gene set, as with the context in which these genes were generated, kind of gives you some other kind of context in which this pathway might be functioning about kind of mediating the effects of cortisol release. Of course, exercise releases cortisol, and then these are giving you specific target genes that might affect lipid storage. And potentially, remember, these are causal effects from the variants on type 2 diabetes. So there's actually a ton of information in that one gene set linkage, right? You have all the information of the genes that are associated. There's tons of stuff known about them. You have the experiment that was done to group the gene set. Then you have the trait. The variants are causes associated with the trait. So each one of these rows in this table has a ton of information. And you could, query them gene set first. You could query them disease first. And this is why, we're very interested in anybody who browses one of, a disease or a gene set. If you see anything of interest, we're really trying to see what can be learned from these, because this is for the first time that some of these mechanisms from CFDE have been cross-referenced systematically with human traits. So give you some other examples. I think Julie will show you that you can query gene set first as opposed to phenotype first, like look for gene sets within a program. And just kind of going through those, another example of MotorPack, like I think the second to top or maybe the second page, is a relationship between upregulation in the blood after exercise with reticulocyte count. Obviously, red blood cell count is well known to have, to be affected by exercise. In this paper I found, most of it's through erythropoiesis stimulation. And if you kind of look at the genes in the top of that gene set, there are a couple that are involved in hematopoiesis, but there are other genes that are involved in like iron transport and structural integrity, which means that those might also be affected by exercise. And I don't know to what extent that's known, but that's just another example that if you're interested in the effects of exercise, this could point you to some other mechanisms that might not be appreciated. Here's an example of LINCs, chemical and inflammatory bowel disease. So all these gene sets from LINCs are genes that are upregulated after treatment with a chemical. So this is a chemical SCH 79797, 239 upregulated genes. It's associated with inflammatory bowel disease. Turns out that compound is not under development as therapy for IBD, but does have antibiotic and anti-inflammatory properties, both of which have been linked to IBD. So not to say that this is like a drug you could use for IBD, but potentially pointing to some type of mechanism that might be relevant to the pathogenesis of IBD, and in particular, specific genes targeted by this compound that might be relevant to it. Multiple sclerosis. The top hit is in GTEx aging. These are downregulated genes in elderly people to younger people, 178 of them associated with multiple sclerosis. It turns out that, and I think the critical thing here is these are in the small intestine. So it does turn out that the intestinal barrier and these genes actually involved in immune system regulation. And so it turns out that, yes, downregulation in the small intestine and the microbiome have been shown to be potentially involved in multiple sclerosis. So again, I'm pointing out this is just the broad level link between the pathway and MS, but here we're getting specific genes that have the genetic variants that are associated. So again, a ton of information there to kind of play around with. And my favorite, because this came from that differential expression that Julie showed, is a gene set. If you look at agent monarchy, these are genes that are downregulated in people with dementia in the hypothalamus of the brain. We're working on getting these Mondo IDs mapped to human readable things. So ChatGPT won't know what this is. But if you do some searching for relationships between dementia and agent monarchy, there actually was this clinical study that showed that either young or old agent monarchy is clinically related to dementia. And of course, the hypothalamus is important to regulating monarchy, right? So that's a potential link between the hypothalamus and dementia and agent monarchy. And if you look at the specific genes involved in them, now this gives you very specific genes that we now know are differentially expressed in people with dementia and also have variants that are causally associated with agent monarchy, which could be investigated to yield molecular links underlying that relationship. And just some very brief searching, actually a couple of genes at the intersection here are involved in appetite regulation, like MC4R, and I think there's another one, I think PCSK1, if I remember, maybe another. But it could be, you could envision maybe there's some brain pathways that control appetite and that affects obesity, which might affect agent monarchy, but then also those same pathways are somehow involved in dementia. I have no idea if that's true, but the point is it's a very testable, not necessarily testable, but specific hypothesis that could be investigated. And one point just that's interesting about this is we've been talking entirely about these marginal effects here, which, as I mentioned earlier, are the effects of the gene set in isolation on the trait. But in this case, it has a very high joint effect, which means that even after you control for all of the other gene sets in the model, this meaningfully predicts agent monarchy, which means it's a completely novel mechanism that is not found by any of the other gene sets that are outside of CFTE. So that's a really quick overview of what we're hoping to roll out more. We're very soon rolling out a lot of rare diseases links as well beyond these 1,200 traits that'll hopefully come in January. And we're very, sorry, this is a slide from ASHT. So we don't have an ancillary event tomorrow. You can look at our webinar if you want to see more about Pigeon. But in particular, I really just want to emphasize there's tons, and I think there's a huge opportunity to leverage the CFDE resources in complex traits. I do not think people appreciate how much information there is in a resource like LINCs or MotorPack, and the people working in that space do not typically study human genetics and vice versa. So I think there's a ton of low-hanging fruit, and all of it's available to be browsed, and we are happy and in fact actively looking for collaborators who are either interested in a trait and see a novel mechanism, or interested in like a CFTE program and are interested in human traits. And, you know, even small findings would be a huge help to us as we work on this functionality. And I really do think could yield to some very low hanging fruit publications linking these for the first time. So Julie, I can turn it back over to you if you want to demo more. We can also take questions if people have them either in the chat or if you want to raise your hand. But Julie, you can take it from here.
That sounds good. And yeah, just stop me if anyone has a question. We can go through those too. So Jason already gave us a flavor of what this looks like, so I'm not going to spend too much time on this to avoid redundancy. But just to reiterate, you can start by searching your favorite gene or phenotype here in our search bar and get to it the way that Jason just described. Or you can get to gene set analyses down here, clicking start here, or through the analysis tab, gene set analyses up on the top menu bar. So if you click on any of those things, that'll take you to a page that looks a little bit more like this. So this is the interface that Jason showed you to look, starting with CFDE program or programs at gene set analyses. So by default, we start by inputting all of the CFDE programs, but you can customize these by choosing either one of these 6 individual programs or combinations of programs. So if you start typing, say MO for, refresh my page. If you start by typing MO, for instance, for motor pack, that should come up with start autofilling motor pack. Yeah, there we go. So you can see motor pack in isolation, results from motor pack and the Knockout Mouse project, motor pack and Glygen, etc., so that you can go through each of these and explore the unique biology associated with them. One other thing you can do on this page that I think is really useful is explore this research method tab. So that's actually a place that you can go to start just to learn a little bit more about what we did here, about the pigeon method that Jason just described, the types of phenotypes that are included, and roughly how to interpret your results. So this schematic gives an overview of the types of gene sets that went into the analysis. They're derived from GTEx, IDG, LINX, MotorPack, Glygen, and IMPC, the mouse knockout project. The types of pairings of gene sets you can get, and the differentially expressed gene sets from our other analysis. You can also get a sense of where the traits come from, so you can look up more about those traits on the Association to Function Knowledge portal. And then learn how these gene sets relate to genes, phenotypes, and mechanisms. All right, so if we go back to our search page, let's say we searched for motor pack. That would come up with a page that looks something like this. This shows a plot of the marginal effect, which Jason mentioned, and the joint effect. So the effect of the gene set in isolation within the model and the gene set relative to the other gene sets in the model. And generally for these, higher is better. Above 1 is typically very good, but you can also get sort of suggestive results that might be interesting for biology, even below those sorts of thresholds. So if you scroll down, then you can get these results in tabular form. So a human readable phenotype, links to the phenotype ID and gene set, as well as the source, in this case, motor pack. So if, for instance, you want to learn more about a specific phenotype, you can then click on this link, and that will come up with a separate page where you can then not just see this result of gene sets for reticulocyte count as a phenotype, but all gene set results for reticulocyte count. So you can get a better sense of the phenotype more globally. You can do the same thing for genes. So if you want to see specific genes within these gene sets, you can click show here and then click here to get to a similar gene page. So you can start with a gene first approach and then see gene sets that are a part of which that gene is part. Jason already went through a lot of this, but I just wanted to reiterate that if you're ever confused about where these data come from or what the scores mean, you can always hover above these column names, and that'll give you a guideline for how to interpret them. And then, let's see, finally, I think those were the other major things that I wanted to go through that Jason didn't get through already. But if you have questions, we wanted to encourage you to reach out to us. So you can always go here to our help section, and that will take you to our help desk email where you can reach out for support. So with that, thank you, and we're happy to take any questions.
Any questions? All right, so please, we encourage you, the easiest way it's a very easy to use tool. Like I said, we think there's a lot of information, so please play around with it. And let us know what you think.
And just a reminder, if there are no questions or anything like that, our next meeting is going to be on January 9th, and that'll be a focus group. So be keeping an eye out for that. And happy holidays, everyone. We won't see you all until then. So hope everyone has a nice holiday break. All right.
Take care.
Thanks,
Thank you.
everyone.
Bye-bye.